Howdy everyone! My final project involved growing and studying trichomes in both arabidopsis and tobacco plants. To begin this project, I did a time-lapse video of tobacco on the top and arabidopsis on the bottom and their growth over a period of 5 days. This project is based mainly on scientific observation rather than traditional experimental procedures. The only methods that were used for this study were 1. Sterilizing tobacco and arabidopsis seeds and planting them in an agar medium. They were then allowed to grow over a period of 5 days while a time-lapse video was taken. After 5 days, the plants were placed on a slide and viewed under several different microscopies as shown shortly. What are some structural characteristics in the development of trichomes in arabidopsis and tobacco plants? The significance of this question is that plant leaf hairs or trichomes serve as an excellent model system to study all aspects of plant differentiation and can provide insight at the single cell level. The study of all the developmental stages of a single cell is the first step towards an understanding of how general cellular processes are integrated during development. After 5 days, the plants are placed on a slide and viewed under several different microscopies as shown shortly. Here we have the images immediately following the 5-day growth period. Shoot epidermal hairs are known as trichomes, a term that is derived from the Greek word trichos for hairs. Trichomes are found in most plants and can comprise either single or several cells and can be secretory, glandular, or non-glandular. The functions that are ascribed to trichomes range from protecting the plant against insect herbivores and UV light, to reducing transpiration and increasing tolerance to freezing. For arabidopsis, after the 5-day growth period, there were still no evidence of trichomes. Arabidopsis plants that were already growing in the lab were provided, and they had the GFP marker McCherry. These arabidopsis plants already had trichomes growing on them, and these allowed me to image the arabidopsis trichomes. Studies suggest that trichomes are often the earliest recognizable structures to appear during differentiation of cultured tissue. Leaves produced early in rosette development lack trichomes on their abaxial surface. Leaves produced later have trichomes on both surfaces, and leaves in the inflorescence may have few or no trichomes on their adaxial surface. The production of trichomes appears to be regulated by age rather than the size of the plant, which explains why my plants did not have trichomes growing on them after 5 days. Here we will have the tobacco imaging immediately following the 5-day growth period. As can be seen, these trichomes are very structurally different from the arabidopsis trichomes. They have rounded tips as opposed to the sharp tips of our arabidopsis. These trichomes had the GFP marker, which fluoresced under the WB cube. A week after initial planting, final images of both trichomes were taken once again. After a week following initial planting, final images of both trichomes were taken. Following are the arabidopsis trichomes on the final day. Nicotiana tabacum, tobacco plants, have short and long glandular trichomes. There is evidence that tobacco trichomes play several roles in the defense against biotic and abiotic stresses. Some glandular secreting trichomes have principal functions of producing pest or pollinator interactive chemicals, which are stored or volatilized at the plant surface. There has also been direct demonstration of the biosynthesis of terpenes by isolated trichome glandular heads. Results suggest that glandular heads are the only site of biosynthesis of these compounds in tobacco. Following are the dark field and phase contrast images. In these images, the glandular structures are better able to be seen. Here's an image where the chloroplast can be clearly seen. According to studies, this is where the terpenes are being produced. Here are the references for the general trichome background and more specific tobacco gland background. I would like to acknowledge Dr. Griffin for the use of his lab, also to Krishna for helping me conceptualize this project and allowing me to work on this outside of lab time. Thanks to all of you for watching.